Good morning, grade 9. Today we'll proceed in traits and probability. But here you will find that I will take 2 pages from 7.2, I think, or 4. I write here the pages exactly. I do prefer to illustrate the material in such a way, okay? We're gonna cover those learning objectives today. We'll use the Punnett square to identify the phenotype ratio within an offspring for a monohybrid cross, differentiate between complete, incomplete, and co-dominance. Here, predict the traits of the offspring using probability. A monohybrid cross involves the crossing of individuals and the examination of one mono character like flower color, pod shape, or the pod shape, or different hybrid traits like red color, white color in their offspring. The Punnett square is a useful tool for predicting the offspring genotype and phenotypes in a mating of two individuals. Okay, those we covered before in our class, but here I want you to know that the Punnett square is a tool. Instead of doing calculations, probability calculation, we are using this simple tool, okay? And here I want to go back when we say we're gonna use the the Punnett square. We are writing above here the gametes. What we are writing exactly in the Punnett square, that's why I'm going to illustrate here now. Here, those RR and WW, it's a genotype for a trait like the here, as we can see here, the phenotype with the flower color. Okay, and here at the top and at the side of the Punnett square, we are writing the gametes. As we said before, the gametes are, or the alleles are separated during the formation of gametes, right? So it will be 1R and 1R, 1W and 1W, and we will see this again here. Again, let's see this again. From a sperm and an ovum. What the probability you have? Let's example this are a brown and blue eye color. Here, the dad have capital B small b and the mom have capital B small b. Here are probability. You don't know which sperm will fertilize which egg. Did you get my point? What the components are, what the allele inside each gamete, sperm or ovum. One have capital B, one have small b, capital B, small b. After fertilization, we have one of those four combination. Either this will fertilize this, or this will fertilize that, or this sperm will fertilize this or that. So how we will do this in an organized way in the Punnett square? Okay, you are writing the gametes, one of the gametes, let's say the sperms here and the over here. Let's say it's a genotype capital P small b, and here capital B small b at the side here, at the sides. Then what you are doing, you are adding them. See here, small p, and then small p, capital P. Small p, capital P. We are using the capital letter first, and here capital P, capital P. That's it. So what you are right at the top and at the side, the gametes. And what inside the Punnett square, the adding of them. How they fertilize each other. Okay? Right. That one we are familiar with. We do it before in our class. But here we will take the same thing, but in different cases. It's called complete dominance, which we are familiar with codominance, incomplete dominance, and those are in 7.2 or 4, as I told you, in page 197, 196 in your textbook. So let's go further on this. Complete dominance, it is the one we know, that we work with before. Let's do it one more time. A cross between a pure breeding tall pea plant and a pure breeding dwarf pea plant. But when I say pure breeding, this means that if I give letter T, okay, if I give letter T, pure breeding, this means that they are same or not same. They are homogeneous. So capital T, capital D, and small t, small t. As tall is dominant to dwarf, and both are pure breeding, their genotype must be capital T, and small t, small t. And then if you remember, I told you, I wanted to pass through those steps, 1, 2, 3, 4, 5, 6, and there's 7 the ratio. Okay, here the parents are tall and dwarf. Those are the phenotypes. The genotypes, the tall is capital T, capital D, capital T, capital T, the dwarf small t, and then you will separate it like a gametes in a circles, like that. Then you will draw the Punnett square, the gametes at the above and at the side, and then you will combine them. Okay, then you will figure out or sum up the genotype of the offspring. And we said F1 it's filial, like for in the Latin language, son or daughter. So it will be capital T small t. How many capital T small t do I have? 1, 2, 3, 4, all of them. So all of them are tall or short? Tall, because the tallness is the dominant trait. Okay, 100%. But if we say a cross between two heterozygous tall pea plant, before I show you, hold the video for a second and guess. Heterozygous, this means they hetero, same or different. So it's small t, capital T, small t, both of them. So both of them will be capital T, small t. Those are the parents. Both of them tall. I put here a wrong one. Let's see if this one. Why I put the same here? Okay, it's right. It's right. Only this word is wrong. Dwarf here, replace it by tall. This one is wrong, okay? So both of them are tall and tall, as he said here, but they are heterozygous, they are different. So the genotype is capital T, small t, capital T, small t, and the gametes will be capital T, small t in circles. Figure it out on the Punnett square, adding them. Yeah, let's count. Here, let's see the genotypes we have. We have capital T, one only. We have capital T, small t, two of them, and one small t. Let's convert this genotype to phenotypes. Capital T tall, capital T, small tall, small t dwarf. So I have the ratio of 3 tall to 1 dwarf. So the ratio is 75% to 25%. Okay? A cross between a heterozygous tall pea plant and a dwarf pea plant. Okay, here. Heterozygous tall, so T, capital small t, and dwarf, recessive, so there's no any chance either, otherwise it's rather than, sorry, to be small t. So it will be like that, the same steps. Start by the phenotypes, tall and dwarf. The genotype, capital T, small t, the dwarf, small t. Split them into gametes, figure it out on the Punnett square, and then start to count the genotypes. Capital T, small t, capital T, small t. I'll count them. Capital T, small t, I have 2 of them. Small t, I have 2 of them. Translated to a phenotype. Capital T, small t is tall, and that one is dwarf, so I have 1 to 1. This means that 50% tall and 50% dwarf. Okay? All that, we took it before. The same thing, the same way how we are figuring out and using the Punnett square to know the probability and the ratios of the offspring when parents are mating here. The same thing we will do in the 2 other cases. But here we have something called codominance and other case called incomplete dominance. In codominance and incomplete dominance, let's show you this. This is the codominance. I want you to figure out what the difference between them. codominance and incomplete dominance. Here, both of them, this flower and the pink one, both of them have parents have the same phenotypes. Yani this one of its parent was white and the other was pink. It gives me an offspring with spotted white flower spotted with pink. But in this case, it gives me pink, all of it pink, not spotted. This is the difference between codominance and incomplete dominance. Codominance, or let's say incomplete dominance, it's a synonym for blending. Blending, intermediate phenotype. Yani here, when the parent is white and the other one is pink, white and red, it gave me an intermediate trait, pink, white and red. Imagine that you are put red painting and above it some of it white. It will give you spotted spots, some white and some pink. It will be blended. It will give you an intermediate color. The same here. Incomplete is blending, but co-dominance, both of them dominant. Like here, both of them dominant, but it's not blended. No, it's to give you both of them appear clearly. They are not blend with each other in co-dominance. Like what? Like you know the cows that we see it white with black patches, codominance. White and black mate or reproduce, giving a white with black patches. Here the same, white flower with with pink patches. Okay, so what's codominance? Sometimes neither of a pair of allele is completely dominant or completely recessive, like the complete dominance we know before, the normal cases we know here. As long as the dominant allele exists, it will dominate the recessive allele. Right? The recessive allele will not appear. This is the complete dominance. But here it's co-dominance. Instead of one of them is completely hiding the effect of the other in a heterozygote, like a complete dominance, they both have an effect on the phenotype. Both of them have an effect. This is called co-dominance. In co-dominance, both alleles are expressed together in the offspring. If we cross a red flower and a white flower, they have a co-dominance inheritance pattern. The offspring would be a flower with red and white patches on them. Patches, huh? Not an intermediate color. Unlike the incomplete one, that we will see it now. Incomplete dominance, where the two parents' phenotypes are blended. Blended is the keyword. Together into new phenotype. In co-dominance, both parents' phenotypes show up together on the offspring. Here the incomplete dominance or blending. In incomplete dominance when there is a blending of two alleles that result in a third phenotype. Here there is a third phenotype, which is pink, that doesn't look like either of the parents. This classic example of when a white flower and red flower are crossed, the incomplete dominance, all the offspring would be solid pink, not patches. A completely new phenotype. You don't see either of the parent phenotype. You will not see a white or red, you will see pink. Two common examples of incomplete dominance in humans are the height and hair color. Yani, if a tall and a short parents reproduce or mate each other, you can find that one of the kids are tall or short or intermediate, right? In between them. Offspring will likely not have the exact same height or hair color as one of their parents, but will often have a blend between the two parents' phenotypes. Inheritance, this for the incomplete dominance, we said the height. Back to the co-dominance. One of the popular examples of co-dominance in humans is the blood grouping. We know that we have 4 types of blood groups, A, B, AB, or O, right? We are familiar that till now, all the traits of phenotypes we talk about is controlled by 2 two alternative alleles only. But the blood group is controlled by three alleles, either A or B or O. A, B, or O. So it has three alternative alleles. Okay? The base letter I, it's called immunoglobulin. It is the, I will show you what the globulin here and what the AB for that. But for back to the genetic side here, that O or IO is recessive. This one is completely recessive. Two IO allele must be present for the person to have type blood O. But so I is completely recessive. Sorry, O is completely recessive. But A and B are co-dominant. What means of co-dominant? If they exist with each other, it will not give me, and A will not dominate B or B will dominate A. No, it will be A, B. Both are dominant to, and both of them are dominant to O. If a person recessive A allele, IA allele, and IB allele, their blood type is AB, in which characteristics of both A and B antigens are expressed. This I'll explain it now. Okay, let's see here. Here we have the blood types A, B, AB, or O. We said that O is recessive. So for a person to be O type, this means that the two alleles are O. Okay, but if it's A, it can be AO or AA, B, BO or BB, AB, AB. Okay, but why we're writing it like I and the uppercase AO and that's it? Because here it's they are codominants. In codominance traits, we are writing it like a capital letter and an uppercase to show the differences between the codominants and the complete dominance. Here, that slide is for this slide is for your information to know what the differences between the blood groups. Here, this is called antigens. What antigens? Antigens, it is the same like how the immune system, our immune system, our antibodies in our plasma, in our blood, recognize different viruses and attack them. And here, let's mention the coronavirus, why it's not, let's say it's a, yeah, it's a health crisis yani for now. Why? Because if in cases it attack or infected a human or individual that his immune system is weak and will not figure it by its own, it will be a critical case. But in normal cases, these antigens, these viruses which have a certain shape or certain shapes or these spikes at its surfaces, it will be recognized by the antibodies and it will attach to it. And if you see here, it's a triangle, and that one is a like a key and lock. Can you see? So here the antibodies in our body can recognize it and break it down, kill it. Okay, this is the same case what happened in blood grouping. That's why when you are donating, when they are transferring blood from one to one, it have to be the same blood group or O in some cases, or AB in other cases. I'll not go in these details, but why? Because if they exist the same antigen, it will cause breaking down of the red blood cells. Okay, so group A, it have this shape of antigens, spikes on its surface. B have that one, and AB have both of them, and O doesn't have. So A will have anti-B, and B will have anti-A, so they not attack each other. Or the blood doesn't attack itself. And in some cases, this happen, and it's a genetic disorder in this case. Okay. Right. Let's practice some of this, like your homework would be. Okay. Yeah. Let's see. Codominance. When I say codominance, is it blending? No, incomplete dominance is blending. So here it will be in between, it will be a color, not spotted, in between our intermediate trait. In some chicken, the gene for feather color is controlled by codominance. The allele for black is B, and the allele for white is capital W. And here, if you can notice, both of them are capital letters because no one is dominant and no one is recessive. Both of them are dominant. The heterozygous phenotype is known as erminate. Without Google it, oh, he mentioned it here, you have to imagine when I say codominant, it's black and white spots on it. Okay? Codominance. What is the genotype for black chicken? What do you think? B. Black. It will be capital B, capital B. طب genotype for white, capital W, capital W. طب genotype for erminate, the spotted one, yeah, it will be capital B, capital W. Okay? A black chicken and a white chicken are crossed. A black chicken and cross chicken. Okay, so a black chicken, its gamete will be capital B and capital B. And a white chicken, capital W, capital W. Okay, yeah, let's figure it. B, W, B. Sorry for being slow. I'm working by a mouse, not a pen. Okay, what the percentage of possible genotypes? Capital B, capital B. Do I have any? Zero. 0%. BW, B capital W, 100%. WW, 0%. The ratio is 100%. BW. The possible phenotypes, black 0, white 0, ornament 100%. Okay, let's go next. This is for the codominance. You will find same one like it, like that one in your homework. Incomplete dominance. Incomplete dominance, incomplete dominance. So what I will remember, blending, it will be in between. In snapdragon's flower color is controlled by incomplete dominance. The two alleles are red and white. The heterozygous genotype is expressed as pink. What is the phenotype of a plant with genotype RR? RR is red. R dash R dash is white. R capital R dash, R dash. What do you think? Is it will be white spotted with red, or it will be pink, incomplete blending pink? Okay, a pink flowered plant is crossed with a white flowered plant. Okay, a pink flower, here a pink, which is R, R dash, and white, R dash, R dash, R, R dash, R, R dash, R dash, R dash, and R dash, R dash. R dash, R dash, R dash, R dash. Yeah, right. Okay, let's see the percentage. I don't have any of R 0. I don't have any red. R dash, I have 1, 2, 2. R dash, 2. So 2, like if we want to make it like percentage, 0 is 0 percentage, 2, so it will be 50%. 2 out of 4, 50%, and 2, half of it R dash, and half of it R. This is the ratio. Okay? Here the same. Right. Here. Phenotypes. Red. Do I have red? Do I have red? R? No, 0. Do I have pink? R dash? Yeah, I do. 50% pink. And white R dash? Yeah, I do. 50%. That's it. So we are following the same rules for you for the dominant, complete dominant. When we using the Punnett square in this case, only the thing that nothing dominant on the other, it will be either co-dominant spotted or incomplete dominant blending. Okay, and that's it. Thank you. Maybe I will upload another video for blood grouping. Blood grouping. Oh, let's see if you can figure it out, or I will upload another video showing you how we do the blood grouping practicing. No problem. Okay. Yeah, let's see. Finish your assignment and submit it before tomorrow 3 p.m. Yeah, let's see you. Have a nice day.